Okay, and now for the exciting part of announcing the winners of our MIXI Teva poster competition of our students. We had 12 finalists that were selected from all of the country, from the different universities. All the presentations and posters were excellent, and I have to applaud to all the students participating. Can you put the slides with their names? We have a slide with all their names, and I know that they are excited here and waiting to hear who is the winner. I also want to thank our judges that were undercover here, going through the posters, asking the questions, and thank you for the students for being patient and explaining so well, and we got many, many compliments. So now I will announce the third place. Not yet, not yet. Wait. The third place winner will give you also a pitch of 3 minutes of the talk, and I want to announce it. The third winner is from the Technion. It's Dr. Avi Rutenberg, to the stage. Avi is also an alumni of our Bio-Innovators Forum. Congratulations, Avi. I don't know if the photographer is here. And Avi, tell us a bit about the research you're doing.
Okay, thank you. So first of all, I'm really excited, and I would like to thank the judges and thank Teva for giving me the opportunity to present my research. And I'm from the field also of cancer immunotherapy, and specifically cell therapy, CAR-T, and other types of CAR-M and CAR-NK, etc., in oncology, especially in solid tumors. And one of the biggest problems that we have when we consider cell therapy in oncology is that we have great results when using CAR-T in blood cancers. But when we consider solid tumors, so we know we have a problem. It's not working well. And we know that currently there's no CAR-T therapy approved by the FDA for solid tumors. And one of the main reasons for this limitation is metabolism. Today we know that the tumor actively competes with the immune cells over nutrients, over essential nutrients. And in solid tumors, we have a problem in the tumor microenvironment of starvation. So the tumor starves the invading immune cells. And as a result, we have immunotherapy failure. In my research, we propose to overcome this problem by harnessing nanotechnology. And in fact, we propose a very simple concept solution. And what we are trying to do is we nano-encapsulate the metabolites that the immune cell needs. So we encapsulate mostly glucose and several key amino acids like arginine and glutamine in the nanoscale. We developed a very high yield encapsulation methodology. So we actually have a core shell nano architecture. We have the nanometabolite in the nanocapsule as solid state. So that's like the highest concentration you can have in the solid state. And it's surrounded with a nanometer-sized silica shell. And what we propose is very simple. We take our food nanocapsules and we feed them to the immune cells, like CAR-T cells, and we focus on CAR-T cells and CAR-M cells. We do the feeding during the ex vivo stage of adoptive cell therapy. So the night before you reinfuse the CAR-T cells or the CAR-M cells back to the patient, you just put our nanoparticles to the medium, the cells uptake it from the medium, and then you get like metabolically enhanced cancer immunotherapy. So I'm not going to get into details, but I can tell you this, and you can look at I will explain on the CAR-M immunotherapy because maybe it's easier to explain in the short time. But we see, you can look at the bottom graphs on the left, we see a dose response. The more nanoparticles we feed to the CAR-M cells, we see a better survival under starvation conditions and even some proliferation in a dose-dependent manner. We have also initial positive results in T cells. And if you're interested, I'll be glad to elaborate afterwards. And I will conclude by saying that while everyone focuses on searching for the next immune checkpoint inhibitor, in our lab we say, think different. The next Keytruda is going to be metabolic. Thank you.
Each one of our winners will also get a 1-year IATI membership. Thank you. Good luck. Okay. Let's go to our second place, and this is from Ben-Gurion University, Yuval Yogev. Yuval, just give a disc on key of your presentation. No, I gave it as a... okay.
Thanks very much. I'll wait a couple of seconds until we're finished uploading and when we'll have it. So first of all, let me start by saying thank you very much. It's a great honor. My name is Yuval. I'm an MD PhD. I'm currently a resident at Safra Hospital Center, a pediatric hospital in Sheba Medical Center. And I did my PhD at Ben-Gurion University under the supervision of Professor Ohad Birk. Our lab focuses on deciphering the molecular basis of rare diseases. And in this particular project that I'm going to present, we started off by trying to decipher, discovering a new type of limb-girdle muscular dystrophy, a sort of muscular atrophy that begins at ages 30 to 40, where the patients, we were discussing a family of 7 individuals who suffered from severe muscle atrophy starting at the age of 30, and beginning with with severe muscle atrophy leading to loss of ambulation and eventually loss of breathing. And most of our patients were chronically ventilated for several years. So can I move it from here or is it from you guys? Okay, wonderful. So you can see here the pedigree, and using several molecular and genetic assays, we were able to pinpoint the mutation to a specific gene that was yet unknown to cause diseases, and this gene was HMG-CoA reductase gene. You can see a lot of different graphs here stating that the variation and the gene were all very highly conserved, and this was the first time that the HMG-CoA gene was implicated in disease. Using CRISPR knock-in and several types of biochemical and biophysical assays, we were able to show that the HMG-CR enzyme was severely affected with 70% reduction in the enzyme capacity and lower and a high Km, meaning that the affinity of the protein towards the substrate was very, very low. Now, at the beginning, we know that HMG-CoA reductase is a major enzyme in the pathway that's known as mevalonic acid pathway, the mevalonate pathway, which is a major component in the synthesis of cholesterol. Now, I was thinking to myself, well, what's the association between a muscle disease and cholesterol? So then I remembered that I was actually studying medicine, and the most widely prescribed drug class in the world, statins, have the most severe side effects of muscle aches, which could ultimately even lead to a phenotype very similar to what our patient was suffering from. And so from here, the association was pretty much clear. So at this point, we had a new disease. We discovered a novel gene that was associated with causing the disease that was yet to be described in human. What we thought to ourselves now, okay, is why not try to actually treat those patients? We know that the enzyme is severely injured, and we know that what our patients are most likely suffering from is a lack of the product of this enzyme. What we did, we took the enzymes from yeast, inserted them into a plasmids, which we expressed in bacteria. And using a bioreactor, we were able to gain access to a broth that was really, really rich in mevalonolactone. And then I studied chemistry, and I was able to purify this small molecule to a level of 97% to 100 degrees. And unfortunately, we were very fortunate to obtain a very unique, we were able to gain a very unique access to actually try and treat our patients. And I would like to just emphasize that this is a drug that was never before used in human patients. So we were able to treat our most severely affected patient using this and escalating in dose protocol, and we were able to show that our patient had almost 200% improvement in skeletal muscle function and almost 400% improvement in respiratory muscle functions. The patient was able to raise her arms, was able to stand on her own with support, was able to feed herself and to feed her grandson, and to breathe on her own for 5 hours straight without her ventilators. But I also told you that that HMG-CoA reductase is the drug target of statins. So we thought to ourselves, well, this drug helped our patients. Why not see if it also helped patients who are severely affected by statin myopathy? And so we did exactly that. We took a couple of mice and injected them with high doses of statins, and the mice suffered from severe muscle symptoms very similar to the ones that our patients suffered from. And we've seen that patients that were treated with both statins and our newly drug made a complete recovery and basically had muscle functions which are completely identical to those of the control mice. And so in this project, we were able to take our patients from a new disease to a new gene to a novel treatment to both our limb-girdle muscular dystrophy patients and our statin myopathy patients as well. And with this, thank you very much. And I'm looking for any collaborations that you might want to.
Amazing, Yuval, and good luck. Okay, and now for the first place, you're probably excited. I want to invite Eran Harari and Yaakov Michlin to join me in giving the first place. The first place winner is from Tel Aviv University. I can tell you she's also a bio-innovator from last year, from 2 years ago, sorry, she's a mentor this year. And I want to invite the first place winner, Alina Brusk. And Alina, you will win also a weekend in a Jerusalem hotel of the Brown
I have to say, it's my birthday today, so thank you for this birthday present. Thank you.
Let's take a picture. Congratulations.
Thank you. Thanks. Okay, so I'll continue with the topic of the session. Basically, my work is focused on CNS, and particularly on glioblastoma. Our whole lab, the Nora Morbinsky's lab, works in glioblastoma, which is a very aggressive primary brain tumor that has a survival of 1 year, even with the available therapies. So one of the challenges of this tumor, actually we have many. We have, for example, a highly immunosuppressive tumor microenvironment, and we also have the issue of the blood-brain barrier, which makes it difficult to deliver drugs to the brain. So what we did was to team up with the lab of Professor Marcel Makhlouf, and they have this amazing technology which are nanoparticles called nanoghosts, which are derived from bone marrow mesenchymal stem cells. What's special about these cells? These cells have natural tropism to inflammation because they're involved in regeneration. And in the tumor, we know we have inflammation, so we expect these cells to target the tumor, to home to the tumor, and the nanoparticles as well. So the first thing we did was to prove that these nanoparticles actually can cross the BBB, and we proved this via an in vitro model. And we also proved this in vivo by injecting these nanoghosts systemically and showing that these are fluorescently labeled. And we showed that they home to GBM tumors in the brain only when there's a tumor present. And if you look here at D, we look specifically more in detail into the tissue, and we see that this homing is specifically to the area of the tumor. You see here the tumor is GFP positive, but the healthy tissue of the same brain of the same mice actually is not targeted by these nanoghosts. So now that we know that they actually target the brain and they can cross the BBB, the opportunity of what we can deliver is actually endless, and I'll show you just briefly 2 different applications. The first one is loading these nanoghosts with a drug that targets glioblastoma. And what we see is that if we give the free drug, it doesn't really have an effect on slowing tumor growth and helping the mice survive. But when we deliver them through the nanoghosts, we do see amazing improvement in survival and slower tumor growth. Here you can see a representative mice. And the other application is we can actually make the mesenchymal stem cells express any protein in the membrane that we want. Here we express PD-1, an immune checkpoint. And when we deliver this PD-1 nanoghost systemically, we see that in our tumors that are highly immunosuppressive, we see an improvement in survival and, again, slowing tumor growth. So as I mentioned, these are just two applications, and also there are many applications beyond cancer. Everywhere where we would have inflammation, for example, autoimmune diseases in the brain, we could also use these nanoghosts. So thank you for listening, and I'm happy to share my birthday with you.